Okay, then, good morning, everyone. Um, as Larissa, I must say I would like very much to be in Bodrum, where the weather must be very nice today. But unfortunately, because of the situation, this couldn't be, but at least we can be online. Um, and I am sure you will agree with me on how important rapid diagnostic testing has been to address the current COVID-19 situation. And they still are very important, and they will be. And of course, we want to make them better, more sensitive, more specific, cheaper, and applicable to a wider catalogue of problems. Then our aim in this research line is to find out how magnetism can help with it. Many of you have these days given great lectures about new magnetic materials and devices, like just the one we've just heard. What I will talk today now about is one way to apply some of them for rapid biosensing. And I will not stop in the specific motivations because you can imagine there are many and diverse. So the idea is to detect a biological target by combining it to a magnetic nanomaterial and detecting the latter with a magnetic sensor. Let me start by explaining how a general rapid diagnostic test works. Most of them are based on the lateral flow immunoassay technique. Lateral flow immunoassay is a paper-supported platform. It basically consists of a long strip of nitrocellulose membrane that acts as a microfluidic platform guiding the body fluid along it. A key element of the immunoassay is the biorecognition molecule. This is, for example, an antibody or that selectively captures the molecule of interest. We print one of them, one test line, across the strip. This will be an antibody that is specific against the molecule that we want to detect. And then we will print a second antibody, not so selective, to capture anything biological that passes by. The target molecule is represented in this picture by a diamond and is contained in the body fluid, which is in the tube. We will pour in this tube the nanoparticles to tag the analyte. With this aim, the nanoparticles are bioconjugated to a second antibody, which is also specific against the target diamonds. When the test strip is introduced in the sample, the liquid flows up. The diamonds are captured, forming the test line, and immunomagnetic separation. It is a technique to isolate the cells or the biomolecules of interest out of the body fluid. This is very much related to what Larissa has just explained about creating a magnetic field to capture the nanoparticles. In this case, we have conjugated the nanoparticles to the target biomolecule, and with a magnet, we will be able to concentrate them. And then we remove the supernatant. And then we have got rid of a matrix which can be complex, like, for example, blood. And also we have the diluted analyte much more concentrated, so much easier to detect. This will benefit any type of detection, any type, for example, optical detection. But of course, additionally, we can make magnetic sensing using the same particles as detection reporters. And which are the potential advantages of magnetic sensing? In the first place, the magnetic sensor can detect the particles in the whole width of the paper, while the optical sensor can only detect what it sees on the surface. Besides, quantifying by optical methods is delicate because there are optical interferences from the ambient light or from the paper itself, which can be stained or wet. And finally, magnetic signals are durable. They do not faint in time as color does. Then, what do we specifically need from our magnetic particles? Of course, the first condition we need to effectively mix the nanoparticles with a biological medium is that they are stable in aqueous suspension and that we can conjugate them to molecules. The key for both things is the nanoparticles capping. The metal needs to be coated by a polymer that provides functional chemical groups. The most used chemical endings for covalent attachment of molecules are carboxylic and amino groups. These chemical endings can be activated to provide electrostatic charge to the particle surface. The electrostatic repulsion among the particles contribute to compensate van der Waals and magnetic interactions that tend to agglomerate the particles and then produce their precipitation. But the most crucial role of the chemical endings is that they can conjugate to the antibodies that later will bind to the target molecule by a selective immunoreaction. At the end of the day, what we have is the nanoparticles that bind only to the desired molecule, ignoring the rest of the components of the biological sample. Then, for magnetic separation and also for magnetic detection, we need the particles to have a large saturation magnetization and large initial susceptibility. Of course, we are speaking about magnets. If they have a strong dipolar interaction, they would collapse and precipitate. Then, also to favor stability, we would like the particles to have a very small or null remnant magnetization, large magnetic susceptibility, and large saturation magnetization. All these requirements are accomplished by particles with superparamagnetic behavior. It has it all. And also superparamagnetic sizes are small enough to interact very well with biomolecules. Let me explain a little bit more about the initial susceptibility, because we will use a sensor that works at radio frequencies. It means that we need the particles to be susceptible at about 100 megahertz, which is not trivial. For this, superparamagnetism is an enormous advantage. In larger particles, in microparticles, the susceptibility decays fast with frequency in the order of several kilohertz. When the particles are superparamagnetic, the thermal energy makes them more susceptible up to much larger frequencies. Their Snell relaxation frequency depends on their size. Look, the smaller the particles, the larger the relaxation frequency we have here at the volume. On the other hand, we can see in this equation of the susceptibility that, of course, it increases with the volume of the particle. So we must get to a compromise. And the optimal compromise is about the size, the critical size of superparamagnetism. That is, for magnetite, which is photons. And we can see here the correlation between the detection sensitivity with an inductive sensor, we'll talk about it later, and the initial magnetic susceptibility, which is represented here. Accordingly, we see a linear correlation, the sensitivity increasing with susceptibility, and the optimal values will find them about 12-14 nanometers. When the particles are larger, like this one, many of the particles in the distribution are already blocked, and this is why they do not work anymore so well. But of course, if we need to have a large magnetic moment, but we are reduced in size to the critical size, then what we can do is make use of clustering or encapsulation to have many particles per molecule. To do this, what we'll do is we start from individual particles of about 10 nanometers, and we can encapsulate them in liposomes. Liposomes are vesicles made of a double lipid layer, and inside they have a hole which is hydrophilic, so we can put borders there. Another strategy is to embed the particles and cluster, for example, in a matrix of black carbon, or like these ones in polyacrylic acid. In all the cases, what we get is that we have many particles per biomolecule, which will increase the dragging capacity. Let me then speak about sensing. Our approach here is that we need a technique that does not compromise the advantages of simplicity and affordability of the paper test. We want something easy to use and cheap, and also we want something portable. So we detect the magnetic nanoparticles through the refraction index by scanning the paper strip with a collection of planar coil inductors that are successively switched. This is what we do at radio frequency. So you can see here, this would be the whole apparatus, which is smaller than a mobile telephone. And we can either mechanically slide the strip on the planar coil, or we can use a collection of planar coils like in this view here, which is our latest approach. In these pictures, we can see the transverse cross-section of an inductor and how the magnetic field intensity, here without nanoparticles, is very much increased when there is a magnetic material on top of the coil. We can measure this effect by detecting the impedance at radio frequencies. And then either we measure the variation of the AC impedance of the coil at a given frequency, close to the resonance frequency, for example, with a vector network analyzer that we can put in a microchip. Here on the left, you can see one of our prototypes. This one is to measure the variation of the impedance, and we slide the paper strip. Here we have a collection of coils, and here we look for the shift of the resonance frequency. And here this would be an example of measurements. When we scan the lateral flow, this would be the test line that produces a peak like this one, and this is the control line. Let me tell you a few examples of application for rapid diagnostic testing for disease screening. Imagine that for any reason your physician suspects that you might be suffering from colorectal cancer. Then they will prescribe you a fecal occult blood test. This is a very sensitive technique, but very unspecific. It can be positive for many benign reasons. And if it is positive, the physician will strongly advise that you undergo a colonoscopy. But colonoscopies are invasive. Many people do not want them, and many just cannot afford them. So what if we had some tool in the middle, some tool regional hospital did, and they advised that we should look for the liquid biopsy as biomarkers for exosomes. Exosomes are vesicles that a lot of information from the cell of origin. For example, inside there is RNA and there is DNA, and at the exosome surface there are proteins. For example, here in green we have proteins CD81, CD9, and CD63, and those proteins on the surface of the exosomes appear in all exosomes, no matter the origin. But if we look into the protein CD147, we will find that there are many recent studies that point to this protein as specific of colorectal cancer. So for our test, what we'll do is we will use CD9 to capture the exosomes in the lateral flow strip in the paper, and then CD63 to determine the total amount of exosomes in the sample, and then we will label CD147 to identify colorectal cancer. This is a sample of the lateral flow strips that we find with the calibrations. This will be a blank, just particles, no exosomes, no human plasma. This is a sample of human plasma containing no exosomes, and this would be with an increasing amount of exosomes. This is a Western blot proving that we have CD63 and CD147 in our samples. And these are the calibration curves. In the abscissa axis, we have the number of exosomes, 10 to the power of 7 per microliter. The curves that you see here in black are the measurements made with the inductive sensor. In color, what you see are the lines obtained with a commercial reflectance reader. This is a desktop device that measures the light reflected by the paper. For the magnetic sensor, you will see this is the magnetic measurement, and these other in colors were the optical measurements. And for the magnetic sensor, you see the linear range is all the range of measurements. This is the range of clinical interest. Above this, really, it is not interesting to look for any more exosomes. For the optical method, the linear range is much shorter. And this is because this method, of course, is saturates. In the test line, we have a finite number of antibodies. When all the sites are occupied, the exosomes that get there must cross without attaching. And of course, the surface layer of the paper will saturate before the whole volume does. For this reason, the optical signal saturates much before than the magnetic signal. Also, the inductive sensor has a better correlation factor, so the real linearity is much better in both measurements with both proteins. And what is even more important also, the limit of detection with the magnetic sensor is much smaller than the limit of detection with the reflectance commercial reader. This means that we can detect a much smaller amount. And these measurements, of course, have been done without any magnetic preconcentration. To all this, we can add magnetic preconcentration. So this technique for exosomes, it looks really promising. We are very excited about these results. The particles we were using here are clusters of magnetite of 10 nanometers, and these are clustered with a double layer of oleic acid. We put a double layer to make the oleic acid coating hydrophilic, and the particles are superfermionic. And these particles proved to be very good for this application for an additional reason. Look at these experiments that were done on the left here in purified sample. So this means that the sample that the doctors give us are human plasma, and we need to ultracentrifugate and filtrate and other washing procedures, a lot of them producing some loss of the analyte. But it works. This test is made with gold particles and this one with magnetic particles. But when we wanted the other experiment made directly in human plasma with no pre-treatment, the gold particles did not work. We can see only the control line, not the test line. While with the magnetic particles, we see both the test line, very much intense, and also the control line. We think the reason is precisely the fatty acid coating that favors a nice flow of all the complex matrix, the human plasma with all the content. So this is excellent news to achieve rapid diagnostics. And I would like to show you now some results on another bioprotection problem in food safety. You may know that foods and beverages that undergo fermentation, either in their production, like wine, beer, or chocolate, or at the beginning of their decomposition, like fish or seafood, they may contain a molecule called histamine. You probably have heard about histamine. Large amount of histamine may be lethal. This is why we must not eat fish when it starts to decompose, because it has large amounts of histamine. But even small amounts of histamine can produce symptoms in people who are sensitive to it, who are intolerant to histamine, and there are many. And this means that maybe you drink a glass of wine and you develop an awful headache. This is called the red wine migraine. So in many countries, the levels of histamine in wine and beer, they are regulated by law. So this is very important for the wine industry and for consumers, of course. I am one of those people allergic to histamine, so I engaged enthusiastically in a project to detect histamine in red wines. And here you can see an example of the magnetic lateral flow that we obtained, different colors or different amounts of histamine. Here on the right, the comparison with magnetite particles coated with oleic acid on the left, and gold nanoparticles like the ones that the commercial tests use on the right. And we can see in the case of red wine, if we try to use these gold particles, they will not be efficient because the color of the wine is the same color as the particles, and the wine very much stains the paper. This is not a problem for magnetism, of course. Then look, here is the validation of our results after all the calibration. We made these tests in real samples of red wine in different production stages. And well, you see the comparison with the paper that is so easy to use, and with the standard technique, which is ultra high performance liquid chromatography, very sensitive, but very sophisticated and expensive technique. The comparison is really, really great. Then my conclusion to the question, can we use magnetism to develop a breakthrough generation of rapid diagnostic technology? I am absolutely convinced that we can do it in this way and probably with other approaches. So we can gather a lot of the magnetics knowledge that we are discussing at this conference and put it at the service of rapid diagnostic testing to develop practical and sensitive technologies to face crucial challenges in health and environment, and also in food control. And with this, I'd like to give credit to our collaborators in these projects, and of course to the funding agencies, and thank you for listening so kindly.